Hi dear students, let's come back through our journey to DNA. With the advent of microscopes, we had found out that single-celled microbes could be seen. But even then, there was a debate in the 1940s regarding the occurrence of genes within the DNA. At that time, two main scientists, that is Tatum and Lederberg, they conducted some experiments and they found that genes could be transferred from one bacteria to the other by a mating process called the conjugation. And later on in 1952, Hershey and Chase also conducted some experiments in viruses, and they showed that even in viruses, when they infect the bacterial cell, the DNA was sufficient enough to infect the bacterial cell. So today we are going to deal about these two experiments, one which was being carried out on conjugation as well as the one which was being carried out by Hershey and Chase to approve or to authenticate or to support the experiments which were being conducted by Avery to say that DNA was a genetic material. So we are going to analyze today what are the experiments that prove that genetic material of bacteria as well as viruses did contain genes. So let's continue. Now we have Tatum and Lederberg, the pioneers who discovered bacterial conjugation. Do bacteria possess any process similar to sexual reproduction and recombination? And in 1946 we can see that Tatum and Lederberg, they studied two strains of Escherichia coli with different nutritional requirements. Strain A would grow on a minimal media only if the medium was supplemented with methionine and biotin, whereas strain B would grow on minimal media only if it was supplemented with threonine, leucine and thiamine. So the experiments were being conducted on two different strains of bacteria to identify whether some gene transfer was occurring between them by a process called conjugation. So let's go into the details of it using the animated videos provided by the DNA Learning Center. So this is Joshua Lederberg. In 1945, he was a graduate student in Edward Tatum's lab, and he came across to read Avery's paper on the transforming ability of DNA. He became very excited by the possibilities, and how DNA was getting from one bacteria to another was a mystery. One possibility is that the bacteria could mate and physically exchange DNA, and this was the bacterial equivalent of sex. And Edward Tatum made mutations in the bacteria of Escherichia coli to further study the one gene, one enzyme phenomenon. Like Neurospora, E. coli can normally synthesize all the nutrients it needs. Now, for example, it has enzymes that could bind and convert the precursor molecules into essential nutrients like amino acids, methionine, proline and threonine, as well as the vitamin biotin. So if you can see, all the genes that are being needed for the synthesis of amino acids like methionine, proline and threonine, as well as a vitamin called biotin was being present in E. coli. Now the mutant strains that Tatum made were in such a way that they had inability to prepare some of these nutrients. Now the first mutant had some genetic mutations in methionine as well as the biotin synthesis, and they were unable to make these amino acids as well as the vitamin, that is methionine and biotin was not able to be synthesized, but it was also able to make all other amino acids and vitamins. So that's what's depicted here. You have the mutant 1 which was being exposed to X-rays, and what happens, methionine was not made and the biotin gene also was getting exposed, and so biotin synthesis was not being done, but the proline and threonine synthesis was possible for this mutant strain 1. On the other hand, now you have the mutant 2 where the mutations were being carried out for the synthesis of proline as well as threonine. And all of the amino acids and vitamins were able to be made by the mutant 2. As it has been depicted in the picture, you can see here methionine and biotin is able to be synthesized, but no proline as well as threonine could be synthesized by these organisms, the E. coli, since they were being mutated by exposure to X-rays. And now these mutant strains, they can grow on agar plates if the right supplements are added to the media. Now, if you are giving methionine and biotin supplements or proline and threonine supplements, of course, we need a microscope to see a single bacteria, but on agar plates, as we know, we can see the colonies because as they grow very fast. So the mutant 1, when you supplemented methionine and biotin, they formed colonies in the first media, and mutant 2, they were being found to be proline as well as threonine negative, but when you supplemented the required nutrients in the media, they were also able to be grown and they multiplied. Now what happens again? All the components of methionine, biotin, proline and threonine was put in the media, and what happens, you mix some cultures of mutant 1 and mutant 2 was being put into it. Since mutant 1 could make what mutant 2 needed and vice versa, he took these two mutants to test for genetic exchange. First he mixed the two strains and then he grew them together in a culture media containing all the 4 supplements. And after the two strains had grown together for some time, he spread them onto a culture plate with no supplements and let them to grow overnight. And the spreading techniques isolate individual. The spreading technique was mainly used to isolate the individual bacterial cells. And any cell must have all the genes needed to make all the 4 nutrients because they had to survive in a media where no supplements were being given. And the survivors would reproduce and he would see it on the visible plate. As he hoped, he can see that when he examined the plate after the following morning, he could see that a few bacterial colonies had grown on the unsupplemented plate. So, when the two strains were being grown together in this particular conical flask, which contains all the supplemented nutrients like methionine, biotin, proline and threonine, they were able to grow. Now, mutant 1 required methionine and biotin, that was provided, so it was able to grow here. And mutant 2 was in fact an oxytop of proline and threonine, but in this media we had given proline and threonine. So, both of them were able to grow in this conical flask, supplemented with a media as well as other essential amino acids. But when they were being further plated onto a plate which lacked methionine, biotin, proline and threonine, he could see that both the strains were being found over here. That indicated that they had regained their ability to synthesize methionine, biotin, proline as well as threonine. So this could be possible only if they are transferring between themselves something, and that was being possible due to the presence of the process called conjugation. I guess this is clear to you. So that's what he saw. And the only way that any bacteria could grow on this unsupplemented plate is if one mutant had donated a copy of genes to the other, and he calculated that this exchange occurred about 1 in every 10 million bacteria grown together in the flasks, and he named this process of gene exchange called the conjugation and believed that it occurred through direct contact between bacteria. So this is the discovery of conjugation. We would be continuing with the discovery of virus transfer of DNA into the bacterial cells during their infection, in Hershey-Chase experiment in the next video. Thank you for now.